We start by introducing GEO Database. The full name of the GEO database is Gene Expression Omnibus, which is a gene expression database created and maintained by the National Center for Biotechnology Information. It contains microarray sequencing data and high-throughput gene expression data submitted by research institutions in various countries. Most of the published papers and the gene expression detection data involved in the papers can be found in this database. So how do we enter the GEO database to retrieve and download data? We can access the GEO database through the official website. We open the Chrome browser, type the URL, and enter GEO database. Similar to PubMed, the GEO database search page also has a search box. We can enter the name of the disease to be studied in the search box, just like a Google search. We can also add other keywords for an advanced search, such as disease-related tissues, name of the cell lines, etc. Here we take diabetes as an example. The GEO database interface includes search box, search result list, search result restricted options, search record information, etc. The left and right sides here are the search result restricted options. To accurately find the required data, the qualification conditions are very important. The entry type on the left is to restrict the data record type. First of all, there are 4 types of data import: datasets, series, samples, and platforms, which start with GDS, GSE, GSM, and GPL, respectively. Among them, GPL is the platform used for expression detection, in other words, the detection method, whether it is a gene chip or a protein chip or high-throughput sequencing, etc. If it's a gene chip, we can also know which company's chip it is. GSM is the expression data of a single sample. This sample may be a sample in the gene chip, but it can only be data of a single platform. GSE is a series of data for the entire research project, which may involve different platforms, and it is also the most commonly used. A GDS corresponds to the data set of the same platform. Here GSE and GDS will contain multiple GSM data information. Study type here is the restricted test type. It should be noted that different experimental test types are different, such as DNA, mRNA, SNP, and methylation. When searching, you must select the corresponding test type based on your own experimental design. When you want to retrieve the corresponding data of an article published by a specific author, you can add the name of the author here for precise retrieval. Top organisms is the source of restricted species. When selecting the restricted conditions, it is also very important to select the source of the species of the test sample, such as human, mouse, rabbit, and dog. In addition to different species, if you want to find data on gene expression at a certain level, such as tissues and cells, you can add the desired type here. Now we will briefly introduce the interpretation of the search result interface by taking the keyword as diabetes, the filter condition as series, the detection type as expression profiling by array, and the species as Homo sapiens. In the middle is the basic information of the search result list. You can see that there are many results. We click on one of them to take a look. The title is the name of the experimental design. Below are the species type, detection type, as well as the design summary and plan of the experiment. The following is the author's contact information and method, and whether there is a publicly published paper for this experiment. The following is the information of the testing platform and the design information of each sample. This is GEO2R, a plugin for online analysis. In the next lesson, I will show you the download of different data formats and original data. We have finished introducing the GEO database. You can try to search for relevant keywords by yourself and experience it. In this course, we will use the GSE 1009 data set as an example.